For our next series of lecture topics, we're going to shift to talking about how cells grow and reproduce. In other words, how cells organize their activities through time. There are a lot of different components to how cells do this, so it's going to take us multiple lecture videos to thoroughly tackle this topic. We're going to start out here by talking about a cell's life cycle and how cells reproduce, and then we will talk more specifically about how it is that cells transmit information from one cellular generation to the next. So we'll start out by talking about the biggest division in a cell's life, and that is the division between growing versus reproducing. If we look at any given cell at a certain point in time, a given cell is typically only going to be doing one of these two things at any particular point in time. So only growing or only reproducing. So let's start out for a moment by talking about what might need to take place while a cell is growing versus while it's reproducing. So take a moment and think about these questions. What kinds of things do you think a cell needs to do while it is growing? And what does it actually mean for a cell to grow? You might want to start thinking about different organelles while jotting down some potential answers. My next question is, what kinds of things does a cell need to do to get ready to reproduce? Okay, so let's take a moment and jot down some lists to think about these questions. Okay, so for growth, what that means is that a cell is going to be getting physically larger, right? So if the cell is getting physically larger, what that means that it's going to need things like more cell membrane. And that might also mean that if it needs more cell membrane, it's probably going to have to build a larger endoplasmic reticulum, for example. And doing all of that work is also going to require more energy. So a cell might also need to build up its collection of mitochondria. Okay? And so, there's just a couple of ideas of some of what might be involved in cell growth. And it's probably just in general also going to increase the amount of proteins that are present in the cell. And since, the proteins are the main molecules that get work done inside of a cell. Okay, so now in contrast, for thinking about cell reproduction, what typically needs to happen is we need to go from one bigger parent cell to two more or less identical daughter cells and these daughter cells should be structurally and functionally similar to both the parent cell and to one another. Each of these new daughter cells is going to need to get about 50% of the parent cell's organelles and cytoplasm, right? And one of the most important pieces of this process, though, is that both of these daughter cells need to inherit an exact replica of the hereditary information of the parent cell. So this relates back to that last component of cell theory, right? For them to be identical, they have to have identical information in the nucleus. There's one thing about growth and reproduction, though, which is that cells need to use DNA for both of these jobs. So illustrated here on the left is a set of enzymes that are involved in the process of DNA replication. So that's the process of copying DNA, making two identical copies in preparation for cell division. And over here on the right is an enzyme that's called RNA polymerase. And this RNA polymerase enzyme is responsible for transcribing the message from DNA into RNA and eventually proteins to do the work in the cell. The problem is cells need to do both of these things, but they can't do them at the same time, or they'll have a whole lot of problems, right? Because these are both using a DNA, and they're using DNA in different ways. So if they bump into each other, they're going to have a lot of problems, and you could wind up with a real traffic jam inside of the nucleus. So we're now going to take a look at how cells manage these projects. And we're going to start out by examining the prokaryotes, and then we'll move on to take a look at the eukaryotes. So in prokaryotes, the process of replicating DNA and cell division actually happen at the same time, in large part because the prokaryotic DNA is a lot simpler than eukaryotic DNA. Prokaryotes just have that single circular chromosome. So what that means is that the prokaryotic cell's life cycle is generally broken down into just two main components. So there's this B phase up here, which is when cell growth happens. And then there's the C plus the D phases, where the DNA starts to replicate, and then where the cell finally pinches in two. So note that the B phase stands for the birth phase, and then the C and D phases were just labeled that way because C and D come after B. So kind of a random naming scheme. But in any case, during the C phase, that's when the DNA in a prokaryotic cell will start to replicate. So I'm going to draw out a picture of what that looks like. Okay, so to start out with, here is my prokaryotic cell. And prokaryotes have just a single loop of DNA for their chromosome. And that loop of DNA is actually attached to the cell membrane at one specific point. So it's just illustrated right here. So when this cell gets ready to reproduce, what happens is it will start to copy DNA at this attachment point. So this is also just called the origin of replication. Okay, so it's going to start copying the DNA here. So there's just a little loop where it's just started to copy. And the copy is also going to be attached to the cell membrane here. So we've got two attachment points going on. We've got our original DNA here in blue that's attached, and then we've got the start of another loop of DNA in green that's also attached there. Okay, so then what starts to happen is in this point in between these two attachment points, so right here, the cell membrane is going to start to grow. And so, as that happens, what's going to happen is the cell membrane is going to start to grow. Okay, so it's starting to grow, and what that's going to do is, as this second strand of DNA is getting copied from the first strand, so there's that first strand, there's the second strand, and there's where the cell membrane is getting added. So you notice that's starting to pull apart these two strands of DNA. And as time continues, this second strand of DNA is going to continue being copied, and more cell membrane is going to be added here. And so as more cell membrane gets added, then this other piece of DNA is going to start getting moved further away, right? As if we add more membrane in here, that's going to separate these two strands of DNA from each other. So that's going to keep separating. And eventually what's going to happen is over time, I'm going to get an elongated cell. So we have that first attachment point here for chromosome number 1. And if we keep adding cell membrane between those two attachment points, here's chromosome number 2, right? So we're going to add more membrane here. And eventually what's going to happen is we'll add enough membrane that these two circular pieces of DNA are no longer going to overlap with each other. Okay, and so then now that they're no longer overlapping with each other, it's possible for this cell to go into the D phase. And so at the D phase, the membrane pinches off, right? So instead of adding membrane outward, that the membrane additions are going to start to pinch off here. It's kind of, yeah, that dotted line doesn't really show that too clearly. So membrane pinches off, and then we have two identical prokaryotic cells that are ready to go back to the B phase. Okay, so that's the process of fission in prokaryotic cells. All right, so as I showed you, as this process continues, basically we go from one original parent cell to two identical daughter cells. And notice that some of the other ingredients here in the cytoplasm also wind up getting divided here. Okay, so that's prokaryotes, which is great. But what about eukaryotes? So here's where we hit up against one of those challenges of being a bigger eukaryotic organism. That challenge is the eukaryotes contain a lot more DNA. And the pieces of DNA in a eukaryote are organized into chromosomes, and the number of those chromosomes in a eukaryote can vary. So a chromosome is a piece of DNA. So for example, up top here in this table is a type of ant, a jackjumper, where females have 2 chromosomes and males have just 1 chromosome. And then here's us, humans. So we have 46 chromosomes we have to keep organized. So that is a lot more information to sort out every time you divide a cell up. And if you think that's a lot, well, we should also take a look at some of these other organisms I've highlighted here. So here's a mulberry bush, and this mulberry bush has 308 chromosomes per cell. Sorting out 308 chromosomes sounds pretty exhausting. But then, if we go up here, we've got this tiny butterfly known as the Atlas blue that has a whoppingly huge number of chromosomes. So it's been estimated to be somewhere between 448 and 452 of them. So that tells you there's so many chromosomes that we have a hard time counting how many are there. And, it just makes me exhausted thinking about what it must take to get those chromosomes organized for cell division. So for eukaryotes, the processes for growth and cell division are more involved. And we're going to look at a schematic for how that's typically thought about in terms of the cell's overall life cycle. Okay, so the cell cycle for eukaryotes is divided into 4 main stages. And so I'm going to start over here with a phase that's called the G1 phase. Okay, and G1 stands for gap 1 because it was originally thought to be a fairly boring stage without a lot of excitement happening. So the first gap in action. We now know there's a lot of exciting things that do happen during G1. And the biggest thing is cell growth during this phase. So this is general cell growth. That's where a cell's taking in a ton of nutrients, it's breaking them down, and it's using them to construct new proteins and organelles. So then a cell is going to arrive at the next phase, which is known as the S phase. And the S stands for synthesis. And this phase is characterized by the replication of DNA. So copying, making another copy of DNA. And I want to note here that it's really important to remember that this is a distinct phase in the cell cycle that happens well before a cell divides. So cell division is going to happen down here. Okay, so as we keep going through this topic, the reason I want to point this out is that I often see an error in understanding, or probably just forgetting, where if I ask you later on, so where does a cell replicate its DNA? You might be tempted to say that happens down here during cell division, but it doesn't. It happens up here in the S phase, well before cell division. So DNA replication and cell division happen at very different time points in a eukaryotic cell. So this S phase is then followed by the G2 phase. And in the G2 phase, again, that's going to be followed by cell division. And cell division here, I'm going to illustrate as two major parts. So the first part is going to be mitosis, which is when DNA is divided up. And the second part is cytokinesis. And here the cyto part of cytokinesis refers to the cytoplasm, and then kinesis refers to movement. So that is a division of the cell's cytoplasm and the organelles that are in that cell. And we're going to be getting into mitosis in excruciating detail very soon. There's one more set of names to talk about here. So overall, the period when a cell is dividing here is referred to as the M phase. Okay, so this whole phase here is the M phase. Okay? And then the period where a cell is not dividing, so where it's doing other things, so all the way around here, this is referred to as interphase. So in between the M phase. Okay. So the next question to think about is, so how does a cell stay organized in terms of what phase it's in? And the regulation of the cell cycle happens through a set of molecular control systems. And the way these work is that they're referred to as checkpoints. So that's where a cell will reach a checkpoint, and then there have to be certain molecular conditions inside that cell before the cell can proceed to the next phase. So advancement through the cell cycle is controlled by checkpoints. Okay. So how do these checkpoints work? They're regulated by internal signaling molecules, and the internal signaling molecules are only going to deliver a signal to go ahead if certain molecular conditions are present inside the cell. So that sounds really abstract. So let's take a look at 3 sample checkpoints just to give you some examples of how these work. And I think when you see these 3 checkpoints, you're going to be able to start imagining that cells can have other checkpoints as well. But we'll start with these three just to give you an idea. So the location of the three checkpoints we're going to talk about is there's one that occurs here at the end of the G1 phase, and then there's another one that occurs here at the end of the G2 phase, and then there's another one that occurs down here during mitosis, the M checkpoint. Okay, and so let's take a look at how these checkpoints work. Okay, so basically for the G1 checkpoint first, what this cell does is it asks itself, are we ready to go ahead with DNA replication? Okay, so if all the right resources are in place, then the cell will say to itself, yes. And then it will shift its activities over into DNA replication. So it will enter the S phase. Okay? So basically it releases a go-ahead signal. You know, it's like the starting gun at the start of a race, right? So go ahead, get underway with replication. And so that says, okay, keep going with DNA synthesis. But now if that cell is not ready to go ahead with DNA replication, or it shouldn't go ahead with DNA replication for some reason, that is where there can be an interesting branch point. So a cell that isn't supposed to continue with DNA replication can actually switch over at this point to another stage, which is called the G0 phase. And in this G0 phase, the cell stops growing, or growth is arrested. So it's arrested or paused. Okay. So let's think about, for example, let's say you're growing some skin cells, very busy growing skin cells. So as you get larger, your skin also needs to get larger. And part of that growth is going to happen by going through this cycle, cell growth, cell division. But once you stop growing, your skin cells don't really need to keep growing at the same rate. So some of your skin cells may hit this checkpoint and they might enter the G0 state instead. So then sometimes a cell that's in the G0 state might need to start growing again. So say, for example, you experienced an injury. Okay? When you're injured, part of the process of healing from an injury can involve the release of some external growth factors or external growth signals. Okay? And those growth factors will send a signal to the cell to switch back out from the G0 state and back to the G1 checkpoint. And there it will wait until it has all the things are in place, and it will then issue itself a go-ahead signal to head into DNA synthesis. Okay, so that's the G1 checkpoint. Pretty cool. So a cell can go in two different directions depending on conditions that are present. One question that I want you to ask yourself is, why do you think, and let's write this out, so why do cells enter this G0 non-growth state from this point here at the G1 checkpoint instead of entering it at some other stage? So why do cells enter the G0 state from the G1 stage instead of some other stage, e.g. SG2M. Okay, so that's our question here. Got this question written down, so you can come back to it and think about it later on after we talk about some more things. Okay, so that's our G1 checkpoint. So moving on, so here is the G2 checkpoint. At the end of the gap 2 phase, so down here, right? Okay. And this is a stage where a cell is going to check and make sure that it has everything it needs in the right place to begin cell division. So, am I ready to start cell division? Okay. And again, there's two possibilities for a cell at this point as well. So one possibility is maybe something not quite right happened over the course of this S phase, right, where DNA is getting replicated, or something during the G2 phase when the cell is preparing other bits of machinery for cell division. So maybe some of the proteins that get made during the G2 phase weren't manufactured quite right, or maybe there's some mistakes in the new copy of DNA that got created during the S phase. So if the cell detects that these kinds of things have happened, there's some kind of damage to all the supplies and the equipment for cell division, the cell is going to pause here to let those repair processes catch up. And it could also be some of those processes take longer than others. So some processes are ready to go, but others aren't ready yet. It's like leaving on a camping trip. I'm always ready to go camping, 45 minutes before my parents are. And so, I'm in the car ready to go, but we can't leave until my parents are also ready to go. So, if we reach that checkpoint, I'm going to be sitting there waiting until, the car is completely packed up. We've got food, we got sleeping bags, and we're totally ready to go camping. Okay, so if anything of that nature happens, so you can reach that point, and the question might wind up being no. Then the cell is going to pause at this G2 checkpoint to allow time for those processes to catch up. So then, if that happens, it's going to wait, and then it's going to come back and it's going to ask itself again, am I ready to start cell division? Okay, so then if from there the answer shifts over to yes, then the cell is going to give it the go-ahead signal. Okay, you can go camping, or in this case, you can go and divide to cell division. Okay, so proceed to cell division. Okay. All right, so that's the G2 checkpoint. Okay, and so now the last checkpoint we're going to talk about here is the M checkpoint. And this checkpoint occurs during a specific stage of the process of nucleus division known as the metaphase stage. And we're going to be talking much more extensively about this process of mitosis that happens here really soon. So for now, the main thing just to note down about this M checkpoint is just the question that a cell asks itself at this stage, which is this question. Are the chromosomes all attached to the spindles and at the metaphase plate. So if all the chromosomes are not ready to go and are not at the metaphase plate, then the other chromosomes are going to wait until everything's there ready to go before moving forward. So again, the full meaning of this question is going to start becoming more clear once we talk more specifically about mitosis. But for now, that's the eukaryotic cell cycle, all in one glorious picture. So it's a cycle, so that means it's a continuous process that can keep going and going and going. Or as we've seen, it can also stop if a cell reaches that G1 checkpoint and the signs and signals all point towards the idea that maybe that cell should enter G0 instead. Okay, so now in our next video, we'll start to examine mitosis, so that process down here, and cytokinesis in much greater detail. But before we're ready to dive into mitosis in all of its glorious detail, we'll spend some time looking at how DNA gets organized in eukaryotic cells. So hopefully, in thinking about this, you'll recall that one of the challenges that arises for eukaryotic cells with their increase in size is that challenge of keeping all that extra information and all those extra instructions organized. And the solution that eukaryotic cells have evolved is pretty sophisticated and elegant, and it's quite different from the extremely simple methods used by the prokaryotic cells that I showed you towards the beginning of this video that just undergo bacterial fission and then they keep moving on with life.